Okay, I think, can you hear me? Okay, good. All right, so we'll get started, and I'm very happy to introduce Sarah Keane, today's speaker. Sarah is coming to us from the University of Michigan in Ann Arbor, where she is an assistant professor of biophysics and the William R. Rausch Assistant Professor of Chemistry. And Sarah did her PhD in chemistry at the University of Indiana with David Giedrock back in 2012, working on coronavirus replication, of all things. Probably a lot more popular nowadays, yeah. And after that, she went to go do a postdoc at the University of Maryland in Baltimore County, Michael Summers, which is where I really became familiar with her work. She did amazing work, determined the structure of the HIV genomic RNA packaging element, and that RNA structure kind of set a very high bar for what one could do with NMR and biological polymers at the time, because it was very large, very informative, and was an article in Science. And that's where I just thought, wow, that is really cool. Beautiful work. After that, she started her own lab at the University of Michigan in 2017. So let's see, if I do the math, and you were a postdoc for what, 4 years, and then started your own lab. And Sarah's won just a bunch of awards. I'll just name a couple. She's a Cottrell Scholar. She is a Pew Scholar. She's an NSF Career Awardee. And she is also, I think, just a really super nice person and fun to talk to. There's a friend of mine who even borrowed some bench space in my lab for a little bit. And so today she's going to tell us about one of the things her lab is working on. She's been doing a bunch of things related to RNA biochemistry, but today we're going to hear about the microRNA biogenesis. Welcome, Sarah.
All right, thanks, Sam. And thanks everyone for coming. I'm really excited to be able to share some of the work that my lab is doing. So just a primer on things my lab cares about. So my lab's really interested, if I tried to tie all of our projects together, it's with this theme that RNAs are really important in controlling gene expression. And we want to understand sort of the structural basis for that. So just like proteins, RNAs adopt complex three-dimensional structures that are important for function. But it's not just structure we have to think about. We have to think about the dynamics of the molecules, how RNAs interact with other biomolecules. I have proteins listed, but really we're thinking about RNA-RNA interactions, RNA-protein interactions, and RNA small molecule interactions. And then again, really thinking about how all of these factors contribute to control in the cell. So the projects in the lab are focused, again, kind of broadly. The one I'll tell you about today, or the two stories I'll tell you today, are mostly related with cancer. Okay, so this is the only background that you need to know. Apologies if you have an RNA background. If you don't, it's great, because this is really the only thing you have to know to kind of follow the talk. So microRNAs are short, single-stranded RNAs that are really important for controlling gene expression in the cell. They bind to mRNAs, typically in this 3' untranslated region, and basically control whether or not that message gets expressed. Okay? But this functional element, this single-stranded 22 nucleotide RNA, is not made as such in the cell. It's made from a much longer transcript that undergoes different enzymatic processing steps. Okay? So the microRNAs are initially transcribed into a primary microRNA. The primary microRNA is cleaved at this basal junction by an enzyme called DROSHA. That generates a precursor element. The precursor is actually exported from the nucleus, where it undergoes a second processing step. Here we're processing off the apical loop, this terminal loop region, by an enzyme called dicer. Okay, that generates this duplex. One of these strands gets loaded onto Argonaute to form the RNA-induced silencing complex, and that's sort of your functional microRNA. So because microRNAs are important in controlling gene expression, their levels have to be really strictly maintained. Dysregulation of microRNA levels is very closely associated with many different disease states. And so what we really want to do is think about the mechanism for regulation. And I'll tell you sort of a story in two parts. So apologies, because there's two sets of systems. But again, we're asking questions about how this process can be regulated, and specifically this stage of the process for this talk. Okay, so the first part of the talk will deal with proteins. Now, normally I don't like to talk about proteins. I was talking with Riley, right? RNAs are the cool biomolecules. But in fact, proteins are important, and proteins are actually well-known regulators of microRNA biogenesis. Probably the best known example is that of Lin28, a protein that binds to at least some, and again, this is a different story in the lab, pre-Let7 microRNAs to actually promote the degradation of that microRNA element. Okay. Proteins like SRSF3 are also well known to help localize DROSHA, one of the processing enzymes, to the right part of the primary transcript in order to facilitate proper cleavage. There's been some pretty recent mass spec work done, proteomics done, to identify a whole bunch of different proteins that bind to and regulate the biogenesis of a series of human microRNAs. And then somewhat related to what I'll talk to you about today is the binding of a protein called HNRNP, A1 in this case, to a primary microRNA called 18A. And what was known is that the binding of this protein actually remodels the loop of that primary microRNA and facilitates the DROSHA-mediated processing. Okay? So what I want you to know is regulation is important, and proteins are one mechanism by which regulation of the biogenesis pathway can occur. So the system that I'll talk to you about is called oncomere-1. This is a polycistronic primary microRNA. This is actually a large part of what my lab is studying. I'm going to tell you a sliver of that today. But oncomere-1 is such named because it's enriched in a number of different cancers. And what's interesting about this polycistronic transcript is that it is not processed in a uniform manner. The processing of this primary element is highly regulated. So you don't get the same amount of 92 as you do 17. Okay? Now these are the levels if you do in vitro processing assays. If you do this in cell, you actually get different levels. Okay? Now the other thing about this transcript is as it's drawn here, it's like beads on a string, independent elements that are sort of joined by linkers. And if that's the case, you could imagine that shuffling the order of those elements shouldn't affect processing. But in fact, if you make a chimeric transcript where you've shuffled the order, you actually also get different processing levels. So this is just all to say that we expect that the structure of this primary microRNA is actually very complex, and that there are very likely protein binding partners that are important for regulating this processing. So we know, in fact, the secondary structure, at least by chemical probing of this RNA, and it's now displayed on the screen. Okay, so you can see all of the different microRNA elements are color-coded, kind of splayed out, but there's lots of really rich multi-helical junctions. Again, the primary nature of this is a subject of a different talk, but what I want to tell you today is that HNRNPs are a family of proteins that have been documented to impact microRNA biogenesis. So the first evidence was really with this precursor microRNA 18A element. Okay, it was identified as a binding partner for HNRNP-A1 by a cross-linking immunoprecipitation experiment. And then more recently, it was identified that, A1, like I said, binds to and remodels this apical loop to facilitate the drosha-mediated processing. What I'll tell you about today is our work on 20A, which is in cyan here. Again, the proteomics work suggests that H and R and Ps bind to 20A. And in fact, if you knock down H and RNP A2B1, so a slightly related family member, in some cancer cell lines, you actually inhibit the processing of 20A. So this seems to nicely link this element, this RNA element, and the H and RNP family. So just, again, very briefly, H and RNPs are actually a much bigger family than I'm showing you here. I'm showing you two family members that we've dealt with in our lab, H and RNP A1 and H and RNP A2B1. They have a very similar domain organization. They've got an RNA binding domain called UP1, or the RBD, RNA binding domain. Both of these contain two RRM domains, RNA recognition motifs. Okay? So what do we know about the family? This is just a very large family of RNA binding proteins. They are implicated in almost every aspect of RNA biology, and linked, again, to a number of different cancers. Okay, so I won't talk about this, but we've solved at least a preliminary structure of 20A in the lab, and it's shown here by NMR spectroscopy. But what I really want to talk to you about is our protein binding work and our enzyme processing assays. So I'm showing you the secondary structure of 20A. Here, what I've done is I've highlighted in blue the two consensus sequences for H and RNP binding, a UAG motif. Okay? It's a very simple motif. Really, you only need the A and the G anyways. So if we wanted to first ask questions about where H and RNP A2B1 binds to this RNA, we can make two different substitutions. We can make substitutions that break the consensus sequence in the stem, and we can make mutations that break the consensus sequence in the apical loop. Okay? If we then do some isothermal titration calorimetry experiments, hopefully you can appreciate that the black curve is for the wild type sequence. The blue is for the mutation in the stem. You can see that these bind almost identically. Okay? However, the mutation in the apical loop that eliminates this binding site has no obvious binding. Okay? So this is pretty clear evidence to us that at the terminal loop, this is the site where you initiate binding. Okay? We've done the same things looking at H and RNP A1, and we see very similar trends. So the next we wanted to look at the RNA itself to see sort of what was happening to the RNA upon protein binding. And so I'm showing you here just a region of an HSQC spectrum. It's a proton-carbon correlation. And we've made a sample where we only are looking at A's and G's. So I've kind of grayed out the C's and U's in this secondary structure. We've also used a truncated RNA just for the simplicity of this particular spectrum. So what we can do here is we can, again, watch how protein binding affects the chemical shifts of all of these different residues. So without protein, we have this spectrum. As we titrate in protein, we can watch some peaks shift and some peaks disappear. Okay? And if we quantify this, at least roughly quantify this, we can see that almost all of the binding, all of the large chemical shift changes are happening in this apical loop. And there's actually no chemical shift perturbations for this adenosine. This is A8 here. We see absolutely no changes. So that's that second binding site, suggesting that this never gets accessed by the protein. Okay, so the next question is, okay, great. So this binds. It's not super surprising. It contains a consensus sequence. But does that binding actually influence the processing of the RNA, the the dicer processing of the RNA? So what we've done here is we've radio-labeled a precursor transcript at the 5' end. And we can look by a gel to see what products are formed. Okay, so we're separating by size. And we're only seeing products that have a 5' end. So we can see the full-length product migrates with some retention speed, I don't really know. And then a truncated product, this is the mature microRNA, migrates differently. Okay? So we can do this as a function of time, plus and minus protein. And if we quantify this, we can actually see a pretty noticeable enhancement of processing in the presence of the protein. So this is actually the first time to our knowledge that we've seen regulation on a precursor element, so that's dicer processing. And the next thing we really want to start looking at is we know that for other elements, other microRNA elements, the H and RNPs influence drosha processing. And so that's kind of our next question is, do we see the same kind of effects or these additive effects when you have both drosha-mediated processing and dicer-mediated processing? Okay, so this is going to wrap up the very first part of the talk. So we've determined a 3D structure for this element. And we've shown that H and RNP A2B1 both binds to the apical loop and enhances the processing efficiency. Something that we're interested in is, this RNA binding domain contains 2 RRM motifs, yet only one site seems to be engaged. And actually, if you look at these 2 RRM domains, they're almost identical. Structurally, they overlap almost perfectly. And so we're wondering sort of what the differences are, if any, between these 2 different domains, and which one perhaps is involved in the engagement. And we're doing follow-up structural biology studies using both NMR spectroscopy and small angle X-ray scattering to look at the complex. And again, we're asking questions about the remodeling of the apical loop structure to enhance enzyme access. Okay, so that's my very quick, proteins are important for microRNA biogenesis. But the next thing I really want to talk about is how RNAs themselves can be regulators of this pathway. So in 2021, so just last year, there was a paper that came out that identified an excited state structure in a precursor microRNA element that modulated the structure. Okay? And it modulated the structure in such a way, it's just a shifting of the register of the base pairing, but it modulated the structure in such a way that it actually also changed the ability for that RNA to be processed by dicer. So this is one of the first examples of really an RNA-mediated regulation for microRNA biogenesis. So this was super exciting for us because we were already kind of asking these questions on our system. So the system I'll tell you about here is called precursor microRNA 31. Okay, it is also oncogenic in nature. It is expressed in a number of different tumors. Sometimes it's overexpressed, sometimes it's underexpressed. And it has sort of dual properties. It can function both as an oncogene and as a tumor suppressor. Okay, so it's a cool system in this sort of delicate balance here. The other thing I want to tell you about 31 is, I mentioned this in the beginning, but lots of precursor microRNAs have known protein binding partners. And this mass spec proteomics table that you can't read, and that's fine, reports on this. What's interesting is of the 72 human precursor microRNAs that were screened in this proteomics experiment, only 2 of them had no protein binding partners. Okay, and 31 was one of those 2 candidates. Okay, so this led us to ask the question, could the regulation really be happening at the RNA level? If there aren't transacting proteins that are coming in and remodeling the RNA, what about the RNA structure itself might lead to that? Okay? So again, I'll just very briefly tell you about these structural features of precursor microRNAs. There are 3 real regions. There's a stem region that contains some number of mismatches. There's a dicing site. This is where the enzyme processing happens. And then there's an apical loop structure. So we actually looked through all of the precursor microRNAs, human precursor microRNAs, and sort of examined how many mismatches were in the stems and what their nature was. And so you can see that the vast majority, 70% of human microRNAs contain at least one mismatch in their stem region. About half only contain one, and the other half contain some number larger than one. Most common, we found CA mismatches, which are interesting in and of themselves because there's some pH sensitivity to this type of a mismatch. And precursor microRNA 31 is predicted, this is just a structure based on sort of thermodynamic principles, of the 31 precursor. And so what you can see here is we have 2 mismatches in the stem region, a 1×2 internal loop at the dicing site, and then an apical loop at the top. Okay. So since we expect the regulation for 31 is happening simply at the RNA level, there's no proteins involved, we really wanted to look at all 3 of these regions of the RNA to ask whether or not they influenced processing. So my lab mostly does NMR spectroscopy to look at RNA structure, and I'm showing you now just a proton-proton nosy spectrum. This is one of our most commonly used experiments of this RNA. I'll point out the size. It's a 71 nucleotide RNA. This is pretty large on the NMR scale. And I'll just again tell you it's a proton-proton correlated experiment. All of these peaks that we see, these are essentially reporting on interproton distances. How close together are 2 protons? If they're close together, we get a peak. If they're far apart, we see nothing. Okay. Now, NMR spectroscopy is great. It's a powerful tool, but it also has a number of challenges. So traditionally, NMR has been applied to the study of small RNAs. You can see this histogram, at least of last year, as of last year, correlates the number of structures as a function of the size of the RNA. Okay? So you can see the vast majority of structures are less than 50 nucleotides. The average size is only 29 nucleotides. Okay, so if you're a protein person, this is a 9 kilodalton protein, right? Not super impressive, right? Okay, and the largest is out here, 155. Okay, and 71 sits here. So it's actually it's a hard system to study, but it's worked out really nicely. We have a few tricks that we use. So the strategies that we use are shown here. So this is what I spent a lot of time in my postdoc working on, is developing how do you prepare samples to get really high quality NMR data, even for these really large systems. And I won't belabor the points here, but you're going to see sort of these pop up again throughout the rest of the talk. Okay. So the first thing we did is we broke the RNA down, the 71 nucleotide RNA, down into smaller components. Okay, so we call these the oligos that sort of make up the whole RNA. So what we can do is we can make an RNA that corresponds to the bottom of this hairpin. We can call it bottom A. And the spectrum here is actually beautiful. It's easy to assign. The data is high quality. We can collect this data and have it assigned within a week. Okay, that's really good for the NMR scale. So we can do for bottom A, we can do for bottom B, just kind of walking our way up the RNA. Small systems that we can then hopefully have a basis. Once we have these chemical shifts, we know roughly where they should fall in the full-length spectrum. Okay? But you can see even for top, which is the top half of this in purple here, this spectrum is getting pretty crowded. Okay? And again, we don't care so much about top or bottom, but we care about the full-length RNA. And so here's the proton-proton nosy spectrum, again, of this 71 nucleotide RNA that's unlabeled. And what I mean by unlabeled, it means that there's protons everywhere. All of the non-exchangeable carbons contain protons. We do all of these experiments in D2O, so all of the nitrogen-bound protons are actually exchanged out. So those are gray. Okay? Now, we're limited here in 2 dimensions. Okay? And we have some number of peaks that we have to resolve. And so our way to do this is actually to go into a 3rd dimension by changing where we have protons in the RNA. Okay? So now instead of an RNA that has protons everywhere, we can make an RNA that has protons only at a few positions. We can make an A2R gRNA. That means adenosine at the 2 position has a hydrogen atom, the ribose, and the guanosine ribose. Okay? So those are our sources of protons now. Everything else is deuterated. And we get the blue spectrum. Okay, so hopefully you can appreciate that this is a much simpler spectrum, right? A lot less crowded, more resolved signals. But this comes at a cost because we're throwing away data for all of the C's and U's and other parts of the A's and G's. Okay? So we have to sort of overcome this limitation by making a bunch of differently labeled samples. So where we move the protons to different positions within each nucleotide triphosphate. Okay? So ultimately, this allows us to complete chemical shift assignments and obtain these really important NOEs, these distance restraints, for structure calculations. Okay, so NOEs, distances are important, but also secondary structure. Okay, RNAs, their primary secondary structure is through hydrogen bonding, base pairing. Okay, and so the first thing we did is we tried to determine the secondary structure by chemical probing. So there's lots of different approaches for this. We worked with Sylvie Ruskin she's now at Harvard, to look at how DMS modifies A's or C's. So this methyl, A's and C's can get methylated at either the N1 or the N3 position when they're not involved in base pairing. A's and C's that are involved in base pairing, those nitrogens are inaccessible to this methylation. Okay, so they stay unmodified, and only A's and C's that are accessible, not involved in base pairing, get a methyl mark. Okay, we can then, through a sequencing analysis, basically monitor how often a particular site in an RNA gets methylated. Okay, and that's just shown here. We've done this in 2 concentrations of DMS, 2.5% and 5%, and you can barely see our background control. There are black bars here for a DMSO control, which just tells us that our mutations are real mutations based on the modification rather than some sequencing artifact. Okay, so this tells us what A's and C's are accessible, okay? And that allows us then to, we can use that information in secondary structure prediction programs to determine a secondary structure, again, with this as a restraint. So what we can see from our in vitro DMS reactivity data is that this apical loop is actually much larger than we originally thought. We have really highly reactive A's and C's in this apical loop region. Okay. Okay. The other thing I want to tell you is that just last year, a in-cell chemical probing paper came out suggesting that most precursor elements are less structured in cell than they are in vitro. Okay. Now this is presumably a structure that's dicer bound, and I'll come back to this sort of throughout the talk. Yeah, anyways, I don't want to give too much away. Well, I guess the next slide's going to give it away. So the truth is we have 2 conflicting secondary structure models. Our chemical probing data says that this RNA has a really large apical loop, really open, it's highly reactive. But our thermodynamic predictions say, okay, this should actually have a cinched apical loop with 3 base pairs formed here. Okay. And all of our NMR data, at this point at least, suggests that this stacking is true. We have really strong NOEs between A40, the H2, and G32's H1 prime, and A41's H2, and U31's H1 prime, so those red arrows. Okay? These are really classical NOEs for ahelical stacking in RNAs. So to sort of get a better idea about the base pairing by NMR, traditionally we would turn to an immunoproton detection experiment where we look for the presence of these pink immunoprotons that exist in base paired regions. The problem with that data for a 71 nucleotide RNA at high temperature, 37 degrees, is there's really not a lot of information here, okay? So we found this to be kind of not very informative. So how are we going to approach this? So what we did is we turned to an experiment called this Best Selective Long-Range H&N COSY that was developed by Michael Sattler's lab, which instead of detecting this exchangeable immunoproton, allows you to basically detect hydrogen or the presence of this base pair by detecting on this adenosine C2 proton. And I'll just remind you, this is a proton we're very good at identifying because of our isotope labeling strategies. So what this experiment does, it's kind of in 2 parts, or I like to think about it in 2 parts. You have the black peaks at the bottom. These are the A to A correlations, the H2 to the N1. So every adenosine in the RNA should be represented by a black peak. And then in red are all of the AH2s to U in 3 correlations. Okay, and again, those can only be detected if you have a hydrogen bond because you push the magnetization across this hydrogen bonding network. Okay, so what did we do? So again, I've got the secondary structure here. Only the A's and U's are shown in black, or the U's that are in base pairs. And the first thing we do is we draw vertical lines, basically from a black peak to a red peak, okay? And if there's a black to red peak, then we can assign it, because we know the chemical shift of this black adenosine H2. So this allowed us to identify almost all of these AU base pairs in the stem, with the exception of this. This actually was not detected. A53 is a tricky base. We can only actually detect this proton if we drop the pH and protonate this adenosine to form an AC mismatch. But that's, again, that's kind of beside the point. Okay, so we have all of these AU base pairs in the stem. We also have all of these A's that don't have red peaks associated with them. Those are the A's that are unpaired in the RNA. Okay? So really the question remains, what's happening up here? And it turns out we have two more peaks that were not previously assigned. Those belong to A40 and A41 here. So this really defines this at least as a transient base pair. Okay? So what other evidence do we have to support this secondary structure? Previously, we did some work on an isolated RNA called bottom B. It contains both of these two mismatches. And we saw that if at high pH, neutral pH, A54, again, that's this adenosine right here, that's part of the AC mismatch, is actually partially protonated, even at pH 7.5. Its pKa is really elevated, okay? And so we sort of asked the question, what are the chemical shift changes between low and high pH for all of the adenosines in this RNA? And we could only get some of them because of just the nature of the spectrum. Sometimes it's a bit overlapped. But what we were able to see, hopefully this color coding makes sense, is that the base paired A's basically have very little sensitivity to the pH of the solution. But the A's that are unpaired, those are the green A's here, tend to have big chemical shift changes upon pH changes. And what I'm showing you here is that A40 and A41, which are these two adenosines, they look more like a base paired A than a not base paired A. They're not super sensitive to pH. Okay, so we think this is just further support for the secondary structure that we have. So with all of this information, we were able to determine the 3D structure of precursor microRNA 31. It's shown here. The color coding is as followed. We've got all of these mismatches sort of color coded in the stem. The dicing site is in yellow, and this apical loop is in like a slate color. Okay, so this is a bundle of the 20 lowest energy structures. The RDCs that we collected are in good agreement with the experiment, and our back calculated, our calculated RDC values, and our sort of geometries are all reasonable. So the structure looks pretty robust. So we then really wanted to ask questions about the role of these different structural elements. Again, we're looking for RNA-only factors, and so we wanted to really investigate each part of the RNA. So first we started with the stem, the mismatches within the stem, and again, we did the same type of assay I described earlier, where we have a radio-labeled precursor element, and we can monitor the formation of this partially processed product and the precursor product here as a function of time with dicer. Okay, so what we did is we made a whole bunch of mutations that either stabilized or destabilized all of these mismatches in the stem. And really unsurprisingly, we found that the stem mismatches really don't affect processing. They moderately affect binding, but really not to any appreciable degree. And this is not surprising based on other literature that exists. Okay? So next we turn to the dicing site. So what's happening at the dicer processing site? Here we made two different mutations. We took our 1x2 internal loop and expanded it to a 2x3 or a 3x4 internal loop. Okay? Now I'm plotting here the processing as a function of time. So you can see that the wild type processes quite efficiently at the 10-minute mark. The single base pair breaking, so breaking this formerly CG base pair, is inhibited, but the double mutation is actually just dead. There's no processing here. So this is actually a little bit surprising to us because dicer has to be able to act here. And so presumably having a little extra wiggle room at the dicing site wouldn't be a bad thing, but it turns out it is. It's actually quite bad to have a large internal loop at the dicing site. So then next we wanted to ask, well, what happens if we restrict the dicing site a little bit? So again, we have our wild type data here. A single point deletion, we deleted this C43. And a double mutation where we both deleted C43 and formed a canonical UA base pair. So basically making a fully paired dicing site. Okay, so the gels are up here. And the first thing we noticed when we looked at the gels is that we totally eliminate the presence of this intermediate product, this partially processed intermediate. Okay? If we then look at the efficiency of processing normalized to wild type, you can also see that both of these constructs, delta 43 and delta 43U44A, seem to be better substrates for dicer processing. They process to a greater extent than wild type. Okay? So seems like restricting the dicing site is actually favorable for processing. Okay? So this allows us to build a model where the cleavage efficiency is something as follows, right? We have a 1-by-1 mismatch. Seems to be better than either a fully paired or the native 1-by-2 internal loop, followed by the 2-by-3, and then again, the 3-by-4 was really quite bad, a bad substrate. Okay, so the dicing site seems to be important. What about the apical loop? So here again, we kind of made a bunch of changes to this RNA. We looked at closing, stabilizing the closing nucleotides of the terminal loop. So we can make either a CG base pair or 2 CG base pairs to really restrict the size of this apical loop. Here, I guess I'm showing you the dicer binding assay. So you can see that these RNAs bind dicer more weakly and are processed less efficiently. Okay. We can also see that, at least for this double mutant, where we've really restricted this terminal loop to be a 4-nucleotide loop, we actually promote misprocessing of the RNA. We actually see the formation of 2 mature bands rather than 1. Okay? So this small apical loop seems to reduce dicer processing efficiency, and the length of this junction between the apical loop and the dicing site seems to inhibit cleavage accuracy. Again, this is consistent with some other literature that's known. Okay, so we shrunk the loop. Now let's expand the loop. What we did is we added non-native nucleotides into the terminal loop of this RNA to make it larger. What you can see here from the binding experiments is that binding is unaffected and processing is only moderately affected. So we're actually making a bigger apical loop is only kind of bad for this system. Okay, so at this point, what we have is a whole bunch of biochemistry that tells us that there are some key regions you might want to optimize if you were, for example, designing shRNAs to silence, to natively make RNAi, or for RNAi experiments to silence a gene of interest. You might want to make sure that well, it doesn't actually matter about the mismatches. You might want to make sure that you're restricting the dicing site, though, and that you've got some intermediate, not too small and not too large apical loop region. Okay? So right now we have guiding principles, but I want to go back to 31 because there's still some information that we haven't really resolved. And that's back to this secondary structure. So the big difference here is, again, the presence of this junction, 3 junction base pairs, or the presence of a large apical loop where those base pairs are disrupted. Okay? So the first question we want to ask is, why are we observing these differences? There's certainly different techniques that are used. They measure slightly different things. They're under roughly the same solution conditions, but not exactly. And then we also want to ask, are these structural differences actually functionally important to the RNA? right? I think that's pretty important to figure out. So what we did is we made a series of mutations where we either stabilized or destabilized the DMS structure or the NMR structure. So for example, to stabilize the DMS structure, we made mutations which forced this junction to be open. Okay. Force the junction to be open. Okay. And we call that 0 base pairs. Or we made a mutation where this GC, or this junction region, is stabilized by GC base pairs. So previously we had 2 AU base pairs. We've made those GC base pairs. So that's GC clamp. We've also made 2 intermediate constructs where we play with the sort of stability of these base pairs. So natively, again, we have 2 AUs and 1 GC. We've made one construct where we have 1 AU base pair and 2 GC, so slightly more stable than wild type, or 3 AU base pairs, slightly less stable than wild type. So by thermal denaturation experiments, we can actually monitor the melting temperature of these different RNAs. And it's roughly what we would expect. Very similar TMs, because again, we're only making or breaking a couple of hydrogen bonds here. But I do want to just note that this 3AU element looks a lot like the 29CAA, so we have no actual evidence that 3AU forms this structure. It might actually just form an open structure. Okay, so we did the same types of experiments, dicer binding. So the first question is, how does the stability at this junction element affect the ability to bind dicer? So wild type is here. If we make mutations that stabilize the junction region relative to wild type, we see an inhibition of binding. And if we make mutations that destabilize the RNA, we see a slight enhancement of binding. Okay? So it seems that a large apical loop structure, this big DMS open apical loop, seems to promote dicer binding. Okay, so what about processing? If we do the same type of experiment using these different RNAs, we can see that mutations that, actually most of them inhibit processing, but mutations with the open apical loop structure really inhibit processing, whereas mutations that are overstabilized still inhibit, but to a lesser extent. This mutation, which is, again, sort of the intermediate, it's a little bit more stable than wild type, but not by much, seems to do just as well as wild type does for processing. Okay? So the stability of these 3 base pairs, right, that's proximal to the dicer binding site, seems to be really important for efficient processing. Okay? So then if we kind of put these elements together, and now I've just ordered the RNA sort of in terms of increasing thermal stability. You can see the TM is here. So we see an inverse correlation between the thermal stability of that junction region and the binding affinity. Okay? Now, if we think about binding affinity versus processing efficiency, we also see sort of an interesting trend. So as the binding affinity decreases, right, the processing largely increases to a point, and then we have a drop. So here, wild type is the only construct that sort of maximizes both the ability to bind and the ability to process the RNA. And we think that this is a function of sort of this, the stability of this junction region. We imagine that neither of these structures is truly correct, and that there's a dynamic equilibrium where you're sampling both populations. Okay? Okay, so I will wrap up there. I will just add to the summary slide. We, again, have these sort of functional rules for designing shRNAs. And on top of that, for 31 in particular, we think that we have a nice marker for this dynamic equilibrium. So RNA structure influences function, the processing of the RNA. Okay? So with that, I will thank my lab. The first story, the H&R&P work, was done by a postdoc, Dr. Liu, actually a high school student who was in the lab over the summer, Luis. And then the second project, the 31 project, the NMR work, was done by a former postdoc who's now started her own position in Slovenia, and then a really talented PhD student, Sitsang Ma, who did all of the very extensive biochemistry. I will say I didn't get to talk about David's work today, but David is an alum of Madison, so thank you for sending him. He's done really exceptional work. And I will thank the funding, specifically Research Corp and NIH for the support. And with that, I will be happy to take any questions.
I was wondering really quick, so you said there's like 70-something precursor microRNAs that are involved in regulating a lot of genes, right? Or
well, there's more than 72. In the mass spec work was only done with 72.
Okay, so there's a couple hundred or several?
There's 1,000 or so.
1,000,
yeah.
Okay. I was wondering if you took that whole collection of sequences, can you take what you know and get predictive information about their processing, or are there any rules that might fall out of just looking
just at the sequences,
gazing or folding them in silico? Like, do they always have like these kinds of patterns, or is there a wide variability?
So there's a wide, and I guess it depends on if you believe or not the thermodynamic predictions, because there's not a whole lot of information on the experimental secondary structure. So yes, I think is the answer. So we have a different system, a LET7 RNA actually, that has a very similar sort of 2 apical loop structures. It's modulated by just a few base pairs coming together and breaking apart. And we can see that stabilizing structure A and structure B either inhibits or enhances relative to wild type. So I think that this is going to become a common mechanism, but I think we just don't know enough about enough precursor elements to figure it out or to know offhand.
So there's no like general pattern where you have a certain apical loop and then a little bulgy thing that the dicer likes to cleave, and you can identify.
Yeah, there are some general trends, but I'd say nothing that's hard and fast across all precursors. Yeah.
Any other? Yeah.
Hi. Regarding DMS reactivity, did you think about doing that assay with a different concentration of DMS? For example, if the DMS is methylating one of the base pair, does it affect the structure to be open so that you're actually seeing more nucleotides getting affected?
Okay, so that's I think I understood your question. So thinking about if you methylate any base, it's actually a permanent modification. You don't demethylate, right? And so that's going to change the ability of that RNA to ever go back to form a base pair. That's what you're sort of getting at?
Yeah, but once it methylates one of the nucleotides, it may open up a structure.
Yes.
That is available from another side.
Yes. Okay, you're 100% right. So so we have these 3 base pairs. If you methylate this A, that's going to get broken. That's going to make these other 2 base pairs also less likely to be paired and more likely to be methylated. Absolutely right. So no, we've only done the experiment at 2.5% and 5% DMS. So we've never looked at lower concentration of DMS to see what happens. But I think you hit the nail on the head. I think like the DMS is reporting on some transiently accessed structure, but once it's open, it's open. And everything else is going to look open to the modifying chemical.
I think for it to work, you have to have less than single hit kinetics for the modification, right? So on average, an RNA is getting hit far less than once per molecule.
Yeah.
And you're only detecting a small amount. So
yeah.
Presumably that's what's going on. But you did have 2 different concentrations, right?
We did have 2 different concentrations where we saw different amounts of reactivity, but the same pattern. Yeah. And we did try to cluster. So like Professor Ruskin has a DREAM clustering algorithm that should be able to separate out different populations if they exist. And we did not get any indication that this RNA clustered into 2 populations. And I talked with Sylvia a lot about this because it really concerned me that we weren't seeing it. But she just said some things don't cluster. You have to have like enough of a difference in the secondary structures to for the program to really pick it up as a new structure. And so we might just not have enough difference. So yeah.
No more questions? Students? Anybody? No? Okay. Well, then let's thank Sarah.
Thanks.